Tickborne Diseases, Confronting a Growing Threat. Catharine I. Pauls, M.D., Hilary D. Marston, M.D., Mile per Hour, Marshall E. Bloom, M.D., and Anthony S. Fauci, M.D. Article. Figures, Media. Metrics. 5 references. Every spring, public health officials prepare for an upsurge in vector-borne diseases. As mosquito-borne illnesses have notoriously surged in the Americas, the U.S. incidence of tick-borne infections has risen insidiously, triggering heightened attention from clinicians and researchers. Common ticks associated with Lyme disease in North America. According to the Centers for Disease Control and Prevention, CDC, the number of reported cases of tick-borne disease has more than doubled over the past 13 years. One bacteria cause most tick-borne diseases in the United States, and Lyme disease accounts for 82% of reported cases, although other bacteria, including Ehrlichia chaffeensis, Anaplasma phagocytophilum, and Rickettsia, and parasites, such as Babesia microti, also cause substantial morbidity and mortality. In 1982, a spirochete was identified as the causative organism of Lyme disease and was subsequently named Borrelia burgdorferi. B. burgdorferi, which causes disease in North America and Europe, and B. pfizilii and B. guarinae, found in Europe and Asia, are the most common agents of Lyme disease. The recently identified B. maanii has been described as a cause of Lyme disease in the upper Midwestern United States. Spirochetes that cause Lyme disease are carried by hard-bodied ticks, see graphic, notably Ixodes scapularis in the northeastern United States, I. pacificus in western states, I. ricinus in Europe, and I. persulcatus in eastern Europe and Asia. B. mamoto, a Borrelia spirochete found in Europe, North America, and Asia, more closely related to the agents of tick-borne relapsing fever, is also transmitted by I. scapularis and should be considered in the differential diagnosis of febrile illness occurring after a tick bite. Patterns of spirochete in zootic transmission are geographically influenced and involve both small mammal reservoir hosts, such as white-footed mice, and larger animals, such as white-tailed deer, which are critical for adult tick feeding. The rising incidence and expanding distribution of Lyme disease in the United States are probably multifactorial, but increased density and range of the tick vectors play a key role. The geographic range of I. scapularis is apparently increasing. By 2015, it had been detected in nearly 50% more U.S. counties than in 1996. Lyme disease's clinical manifestations range from relatively mild, nonspecific findings and classic erythema migrans rash in early disease to more severe manifestations, including neurologic disease and carditis, often with heart block, in early disseminated disease, and arthritis, which may occur many months after infection, late disease. Although most cases are successfully treated with antibiotics, 10 to 20% of patients report lingering symptoms after receiving appropriate therapy. Despite more than 4 decades of research, gaps remain in our understanding of Lyme disease pathogenesis, particularly its role in these less well-defined, post-treatment symptoms. Meanwhile, tick-borne viral infections are also on the rise and could cause serious illness and death. One example is Powassan virus, POWV, the only known North American tick-borne encephalitis-causing flavivirus. 3 POWV was recognized as a human pathogen in 1958 after being isolated from the brain of a child who died of encephalitis in Powassan, Ontario. People infected with POWV often have a febrile illness that can be followed by progressive and severe neurologic manifestations, resulting in death in 10 to 15% of cases and long-term sequelae in 50 to 70% of survivors. 3 An antigenically similar virus, POWV lineage 2, or deer tick virus, was discovered in New England in 1997. Both POWV subtypes are linked to human disease, but their distinct zootic cycles may affect their likelihood of causing such disease. Lineage 2 seems to be maintained in an enzootic cycle between I. scapularis and white-footed mice, which may portend increased human transmission, because I. scapularis is the primary vector of other serious pathogens, including B. burgdorferi. Whereas only 20 U.S. cases of PAV infection were reported before 2006, 399 were reported between 2006 and 2016. Other tick-borne encephalitis flaviviruses cause thousands of cases of neuroinvasive illness in Europe and Asia each year, despite the availability of effective vaccines in those regions. The increase in PAV cases coupled with the apparent expansion of the I. scapularis range highlight the need for increased attention to this emerging virus. The public health burden of tick-borne pathogens is considerably underestimated. For example, the CDC reports approximately 30,000 cases of Lyme disease per year but estimates that the true incidence is 10 times that number. Multiple factors contribute to this discrepancy, including limitations in surveillance and reporting systems and constraints imposed by available diagnostics, which rely heavily on serologic assays. For diagnostic utility is affected by variability among laboratories, timing of specimen collection, suboptimal sensitivity during early infection, imperfect use of diagnostics, particularly in persons with low probability of disease, inability of a single test to identify co-infections in patients with acute infection, and the cumbersome nature of some assays. Current diagnostics also have difficulty distinguishing acute from past infection, a serious challenge in diseases characterized by nonspecific clinical findings. Moreover, tests may remain positive even after resolution of infection, leading to diagnostic uncertainty during subsequent unrelated illnesses. For less common tick-borne pathogens such as PAV, serologic testing can be performed only in specialized laboratories, and currently available tests fail to identify novel tick-borne organisms. Such limitations have led researchers to explore new technologies. For example, one of the multiplex serologic platforms that have been developed can detect antibodies to more than 170,000 distinct epitopes, allowing researchers to distinguish 8 tick-borne pathogens. In addition to its utility in screening simultaneously for multiple pathogens, this assay offers enhanced pathogen detection, particularly in specimens collected during early disease. Further studies are needed to determine such assays' applicability in clinical practice. Non-serologic platform technologies may also improve diagnostic capabilities, particularly in identifying emerging pathogens. 2 previously unknown tick-borne RNA viruses, Heartland virus and Bourbon virus, were discovered by researchers using next-generation sequencing to help link organisms with sets of unexplained clinical symptoms. The development and widespread implementation of next-generation diagnostics will be critical to understanding the driving factors behind epidemiologic trends and the full clinical scope of tick-borne disease. In addition, sensitive, specific and, where possible, point-of-care assays will facilitate appropriate clinical care for infected persons, guide long-term preventive efforts, and aid in testing of new therapeutics and vaccines. In the United States, prevention and management of tick-borne diseases include measures to reduce tick exposure, such as avoiding or controlling the vector itself, plus prompt, evidence-based treatment of infections. Although effective therapies are available for common tick-borne bacteria and parasites, there are none for tick-borne viruses such as PAV. The biggest gap, however, is in vaccines. There are no licensed vaccines for humans targeting any U.S. tick-borne pathogen. One vaccine that was previously marketed to prevent Lyme disease, Lymerix, generated an immune response against the ospalipoprotein of B. burgdorferi, and antibodies consumed by the tick during a blood meal targeted the spirochete in the vector. 5 Nonetheless, the manufacturer withdrew Lymerix from the market for a combination of reasons, including falling sales, liability concerns, and reports suggesting it might be linked to autoimmune arthritis, although studies supported the vaccine's safety. Similar concerns will probably affect development of other Lyme disease vaccines. 5. Historically, Infectious disease vaccines have targeted specific pathogens, but another strategy would be to target the vector. 5 This approach could reduce transmission of multiple pathogens simultaneously by exploiting a common variable, such as vector salivary components. Phase 1 clinical trials are underway to evaluate mosquito salivary protein-based vaccines in healthy volunteers living in areas where most mosquito-borne diseases are not endemic. Since tick saliva also contains proteins conserved among various tick species, this approach is being explored for multiple tick-borne diseases. 5 The burden of tick-borne diseases seems likely to continue to grow substantially. Prevention and management are hampered by suboptimal diagnostics, lack of treatment options for emerging viruses, and a paucity of vaccines. If public health and biomedical research professionals accelerate their efforts to address this threat, we may be able to fill these gaps. Meanwhile, clinicians should advise patients to use insect repellent and wear long pants when walking in the woods or tending their gardens, and check themselves for ticks when they are done. Disclosure forms provided by the authors are available at nejm.org. This article was published on July 25, 2018, at nejm.org. Author affiliations. From the Office of the Director, National Institute of Allergy and Infectious Diseases, NIAID, National Institutes of Health, Bethesda, MD, CIP, HDM, ASF, and the Rocky Mountain Laboratories of the Intramural Research Program of NIAID, Hamilton, Montana, MEB.